Okay. All right, so the first thing we have to talk about is what genetics actually is. And what genetics is the study of how traits are passed on from individual to individual. So we're going to start there. Study of how traits are passed to individuals it is also called, so I'm going to say AKA, which stands for also known as, if you didn't know that, heredity. So compared to other sciences, genetics is really a pretty new science, because remember when we learned about DNA and I told you guys that DNA was like really recently discovered, like in the 50s. So it's pretty new for scientists to like know what genes are and what's causing traits to be passed on. At first, scientists thought that proteins were like the genetic material of living things, but actually, obviously, it's not proteins, it's DNA. So this is like still really a developing science, which is what I like about it, that there's new discoveries happening all the time in genetics, which is cool. Now, I say that, but also the first person who like started working on genetics lived in like the 1600s. He's a super old dude. By old, I mean no longer living. So if we go over here, Gregor Mendel does anyone know anything about him? Go ahead, Lily.
Isn't it like pea plant
Yes.
study?
Yes, Lily said he studied pea plants, and he did. He also was a German monk. So literally what he did is grew pea plants in the like monastery garden and prayed. Like that was his life, and that's it. And he like discovered that traits were being passed like in specific orders to like the offspring of the plants. So he discovered dominant and recessive traits by like messing around with peas, which is amazing. You can be a regular person in your garden and discover things. So this is Gregor Mendel. He's a monk who discovered heredity by studying pea plants. And he's called like the father of genetics because he was the first person who like noticed that traits were passing through generations intentionally. And not just randomly, that like some were passed more often than others. So I'll say here that he like also discovered dominant and recessive traits. Okay, cool. So that's like the intro to the intro. And now we're going to start talking about some of the actual like science words that will help us be able to like solve these sorts of problems and make predictions about offspring. So let's go back over here. First, we need to talk about what's the difference between a gene and a trait. And they're pretty similar. And sometimes they're used interchangeably. But if we want to be really specific, a gene is a segment of DNA that codes for a specific physical quality in an individual. So the gene is the actual like microscopic DNA. You can't see someone's genes, but you can see someone's traits. So their traits are the physical qualities. So this is the physical qualities of an individual coded for by genes. One thing I think I've told you before too, sorry, can you not see? Tell me if this thing is like blocking you, because I think it is. Okay, so like I was saying before, there are so many genes that we still don't know what they do and what they code for. There's also a lot of regions of DNA that are like we call them non-coding regions, which means they like maybe don't code for genes or traits. We don't know what they do yet at all. So there's a lot of discoveries still to be made. Okay. One thing I want to mention before I go any further is phenotype, because it's like the science word to describe someone's traits. So if we look at that, phenotype is the physical appearance of an individual. So an example would be like brown eyes. The way that you tell someone or something's phenotype is literally by just looking at it. That's all you have to do. One time I put a question on a test about this and it was like, how do you tell the phenotype of a dog? And the answer choices were like, made it to another dog and like DNA sequence it and like all these crazy answers. And the answer was look at the dog. Because that's how you tell the phenotype. Okay. Just remember like P for physical. All right, now let's go back over here and talk about alleles. So we've already talked about dominant and recessive, so you kind of already know this, but we haven't talked about the word allele yet. An allele is just a way to say the version of the trait or the gene. So an allele is one version of a trait / gene. And it's represented by letters. So for these Punnett square problems that we're going to get into next time, we'll use letters, capital and lowercase, to represent the dominant and the recessive traits. Whatever we're talking about, we'll use the same letter for each thing. So we'll use like a big A and a small a, or a big B and a small b, or whatever, okay? We already talked about this. So dominant traits are always shown. And we represent them with a capital letter. So I'll use A. And then recessive is only shown when no dominant allele is present. And we use a small letter, little a. When you start to do these problems, I really strongly encourage you to choose letters, because it doesn't matter what letter you use, but it really helps if you choose letters that look really different from each other. Because if you choose like big C and small c, your C's all start to look the same, and that makes it harder. So choose like A's or B's or R's, or like something that looks different each time. Okay, guys, how many alleles do you have for each trait? Yell it to me.
2.
Yes, 2. One from your mom, one from your dad. So I'm going to go back to this chromosome situation here. This is, let's say this is mom and this is dad. You have 2 copies. And on your chromosomes, remember, you have different genes. So let's say this is a gene. This is a gene. Put one here, and we'll put one down here. So what your mom and your dad give you is what determines like which alleles you have. So let's say like mom gave you a big A and dad gave you a small A. And mom gave you a little b and dad gave you a little b. I'm going to D's. Mom gave you a big D and dad gave you a little d. So this determines what you'll look like because you'll look dominant for whatever A is. You'll look recessive for whatever B is because you only have little b's. And you'll look dominant for whatever D is because you have big D and little b. Remember, what's the word for these two chromosomes? They have the same genes? Yeah, homologous. Good job. That's an O. Not an A. Looks kind of like an A a little bit, but it's O. Okay. So now that we've talked about this, we have two alleles for each trait. We can talk about genotype. So down below is the word genotype in red. All right, so your genotype is an individual's two alleles or a trait. You cannot see someone's genotype. So remember, you can see phenotype, but you cannot see genotype. So an example of genotype would be like two B's, where the example of phenotype was brown eyes. So it's like the genes that coded for that brown eyes situation. All right, you have two options. You can either be homozygous or you can be heterozygous. If you're homozygous, you have two of the same alleles. So for example, you could have been two big B's or you could have been two small B's. Either way, you're homozygous. And when you see problems like this, they'll specify for you whether it's homozygous dominant or homozygous recessive. It will tell you. Dominant would be the big B's and recessive would be the small. Sometimes you'll also see homozygous called purebred, especially in animals. So this means that they are homozygous either way. And this is especially used for animals. I'm just putting that on here because if you see a problem that says like, which ones are purebred, then you're like, oh, okay, I know. So typically what that means is like all of the offspring will be two small B's because both of the parents were two small B's. That's why it's purebred, because it's all the same all the way back. And then heterozygous is two different alleles. So this would be like a big B and a small b. Sometimes this is called hybrid. But usually just heterozygous. And then another word that's used to describe heterozygous is carrier, especially when we're talking about diseases. So this is heterozygous, especially used for disorders. So an example of this is like sickle cell anemia. You can have one copy of the gene and not have sickle cell anemia because it's a recessive disease, but you could still pass it to your children. So we would call that person a carrier because they're carrying the trait even though they don't actually show it. All right, now let's talk about Punnett squares. I think most people have probably done a Punnett square before, which is awesome. So what a Punnett square is a chart used to predict the probability of traits showing up in offspring. So probably you have seen this before, but it's usually a 4-box situation like this, where you put the letters that belong to one parent, the alleles, I'm just making this up, going on the side, and you put the other ones going across the top. I want to use a different color, actually. So for this here on the top would be parent 1. This on the side would be parent 2. It doesn't matter at all which one is parent 1 and which one is parent 2. It literally could just be flipped and it doesn't matter, as long as you have just one letter for each box. And then when you fill it in, you just bring the B's across and the B's down. So this B is going to go here. This B is going to go here. Same thing here. One thing that really helps me is to always write my capital letters first. Now you have your answer. So in this situation, what percent of the offspring will look dominant?
Half, 50%.
Sorry. Good job. 50%. So this would equal 50% dominant, 50% recessive. And we'll do a lot more of those later on, but that's just a really basic example. All right, we just have a couple more. If we go back to the side. So sometimes a scientist needs to figure out what the genotype of one of the parents was. So a test cross is a mating to figure out whether a dominant individual was homozygous or heterozygous. And this is done for plants and for animals. But basically you would take your, let's say you had a plant that had purple flowers and you knew purple was dominant. Because of that, you wouldn't know whether the purple plant was 2 big letters or a big letter and a small letter. So what you'll do is you'll mate that plant or cross it, it's the same thing, to a recessive individual. Because then you'll know based on the offspring whether the first individual was homozygous, because all of the babies will have purple flowers then, or heterozygous and then only half will. So it's just a way to figure out what you're looking at, basically. There are 2 types of Punnett squares, monohybrid and dihybrid. Monohybrid looks at 1 gene. That's like the example I just did. It will have 4 squares. Dihybrid looks at 2 genes at the same time. It will be a 16 square Punnett square. And it's really fun. And we'll do some next week. No, after break. But they're really giant Punnett squares. Okay, last thing I want to talk to you about are generation words. So sometimes you'll see different generations abbreviated by these letters. If you need me to go back to anything in a second, I will. So the P generation is the first generation observed. P stands for parent. So it's like the parent generation. The F1 is the offspring of the P. F stands for filial, if you ever want to know. And then F2 is the offspring of F1. So basically these will be the grandchildren of P, which you won't need those today, but you might need them next time when we do some problems. And that's it. Do you have any questions? Okay, cool. Was some of that review? Okay, cool. All right, so the other thing we're doing today, guys, is on Google Classroom.